Welcome everyone to NSTA Engage Spring 21. My name is Heather Ryan and I'm the session's moderator. Chris, Herman, and Rachel Mosey are supporting us behind the scenes today, while Karen Avery and Dan Williams are presenting inspiring student questions with a twist in DNA modeling. Please use the chat to communicate during the session, and then the chat will become part of the recording that will be posted on the Spring Engage site tomorrow. Rachel will post links to resources, registration for your free student modeling pack, and a session evaluation in the chat. I will also monitor chat for your questions, and we will let you know when you can unmute and interact. It is now my privilege to introduce Dan Williams and Karen Avery. Karen has been a biology and microbiology instructor at the Pennsylvania College of Technology for the past 5 years. Prior to that, she spent 15 years in the high school classroom teaching biology, honors biology, AP, environmental, and applied biology. Karen earned her undergraduate degree in biology and secondary education from Lycoming College and her master's degree in biology from Bloomsburg University. She is currently a doctoral candidate in the STEM education at the University of Pittsburgh. Dan Williams teaches AP biology, anatomy and physiology, and marine science, and runs the science research program at Shelter Island High School in New York. Dan earned his undergraduate degree in biology from St. Joseph's College and master's from SUNY Stony Brook. Dan is a past teacher fellow from the Brookhaven National Laboratory's Office of Educational Programs and Cold Spring Harbor Laboratory's DNA Learning Center. Both Dan and Karen have been conducting workshops and trainings on our 3DMD materials for educators like you for many years. They have both served in leadership roles on CBM grants and summer courses. And now they have another outstanding presentation for you today. Let's begin with our program. Welcome, Karen and Dan.
Welcome. Good afternoon, everybody. So as we start out, we want to just discuss some of the goals of things that we are looking to cover this afternoon. Fostering student questioning using dynamic DNA and the double helix student modeling pack. Any good model is going to start, inspiring students to ask questions. What can they see? What can they look at? How can you teach DNA structure? How can you teach DNA function with these kits? What's novel about these kits? What makes them really better than some of the other things out there? And what else can students learn? Even at the end, so I've got a little bit of a teaser of playing with some of these models to teach Photo 51 and Rosalind Franklin's research. The models are that good where we can play into that area.
So just to get you folks warmed up, we have up here some very common models, the kinds of models you see all the time. So I'd like for you to pick your favorite model and use the number to indicate which model you've chosen and tell us why in the chat. So yeah, we have some base pairing action going on here. 4, you can see all the molecules, all the atoms almost. So Brittany's using 2, 4, I like the detail on 4 also. So these results make me think that there's no real perfect model. You probably would use different models for different stages in your teaching, as one person noted in the chat. So as we move forward, I'm going to give you just a couple of details about the 2 models that we're going to be using today in this video.
I'm going to use the 2 base pair set to demonstrate some of the overall features of this kit. You can see here that we've got 2 base pairs. I've assembled an AT base pair and a CG base pair. And already we can see the anti-parallel nature of DNA. The kit comes with extra oxygen caps if you want to cap off those phosphate groups on the 5 ends, and some hydrogens if you wish to cap off the hydrogens on the 3 ends. I'm going to leave those off for now for a demonstration. You can easily separate the base pairs, and you can actually teach deconstructing the DNA molecule, or you can have them construct it. It's up to you. But right here we have a CG base pair, and we can see our sugar, our phosphate, our sugar, and the nitrogenous bases meeting in the middle, sugar, phosphate group. So we already can take a look at the difference between our purines and our pyrimidines. We can also see CG3 is how I ask my students to notice that there are 3 hydrogen bonds between cytosine and guanine. And of course, AT2, here is our thymine base and our adenine base, and easily pulled apart are our hydrogen bonds in the middle. So students will explore that. We can also either construct or deconstruct a nucleotide. And for that, we've got a phosphate group, a sugar, and a base. We'll go into the structures of these a little more later in our presentation. This base is a cytosine, and we're going to attach the cytosine to the sugar. The post on the nitrogenous base matches up with a hole in the sugar, so it's fairly easy to assemble and pretty hard to put it in the wrong place. And then, of course, we've got our phosphate group, which will attach not with magnets, but with a little more robust connector. And that is because those are covalent bonds in between those. And so we don't want them to easily come apart. So now we have our phosphate, our sugar, and our base, which you notice is not static. And you'll also notice that our nucleotide is not flat. So when we move up to the 6 base pair set, you can see that we can talk about, and you can do this with the, even starting with the 2 base pair set, that we have a helical structure here. And in this helical structure, you can begin to see things like the major groove and the minor groove of DNA, and talk about how DNA interacts with transcription factors if you wish. If you want to focus on just the genetic code, or if you want to kind of cancel out the complexities of the sugar and the phosphate structure, we can move to the double helix student modeling pack, which was constructed in a way that would be easy to assemble at home. And it is deceptively simple. So if we untwist our double helix here, we can see that we have 12 base pairs. I've not put all the base pairs in because I wanted to show you some of these features. You can see that we've got adenine and guanine. These are our purines, and you can see that they have 2 rings. And we've got our thymine base pair, or our thymine and our cytosines. These are our pyrimidines. You can see that each one of those has a single ring. You can also see that they are going to form the correct number of bonds. So again, CG3, these guys come together, and AT2. All right, those are fairly easy to put into our overall molecule just by pushing the posts through the foam sides. You can have your students set up a particular sequence if you wish. You don't have to. When we twist this molecule into a helix, you can see that arrows have been added to the backbone so that the directionality of DNA can be addressed. And also you can see it on the end. So you can see we have a 3 end and we have a 5 end. And so in this case, you can talk about the 3 to 5 nature, the anti-parallel nature of DNA, or you can just say that DNA is anti-parallel. You don't have to get into the phosphate end and the hydroxyl end. The coloring in the foam kits are consistent in the bases across this double helix student modeling kit through the FGIK, or the Flow of Genetic Information kit. So that's kind of nice. When we look at our base pairs here, you can see that CPK coloring is followed. So oxygens are red, carbons are gray, hydrogens are white, and so on. That's nice because it corresponds with a lot of the diagrams that you'll have in your teaching resources, such as your textbook diagrams. We're going to go further into each of these kits in a little while.
So DNA gets a lot of airtime, and we rightfully so spend a lot of time on DNA structure, but we can use these models for some important diversions, actually not diversions, these are important things. And I hope to convince you that it might be helpful to your students to use these molecules first to model RNA nucleotides. So I'll show you that in this quick video. I'd like to take a moment to show you how you can help your students bridge the gap between the structure of DNA nucleotides and RNA nucleotides, which is sometimes a question that lingers inside of their brains, but they never really get an opportunity to explore that. Starting off, we have our textbook nucleotide that shows us all the atoms, and it's a good model, but it is flat, and we'd rather go from flat to fabulous, and so let's do that. In order to talk about the structure of DNA nucleotides and RNA nucleotides, we have to focus on the sugar. So I'm going to blow up the sugar here for us so that we can focus just on that sugar. And as you know, organic chemists like to number their carbons. So this sugar reminds me a little bit of a house. And so we're going to take this house and start at the oxygen at the top. And we're going to number each of these points, which represent a carbon. So here we have carbon number 1. Carbon number 1 is really important because if we go over to our original molecule, you can see that carbon number 1 is attached to our nitrogenous base. Then we have carbon number 2, which must be special because I've got it in green. Let's go in here and see what we've got attached. It looks like nothing, but of course that's a hydrogen. That will become important. We've got carbon number 3, which is important because carbon number 3 is attached to that OH group. It is the 3 end of the molecule and will form the 3 end of the elongating strand, which is also important because DNA polymerase and RNA polymerase can only add on to that 3 end. So any incoming nucleotides are going to be added to this 3 end of the sugar with the OH group on it. And then we have our 4th carbon, which is important because it's attached to the chimney of our sugar molecule house. And that 5th carbon, or that chimney, is attached to our phosphate group here. Now that we have our bearings, let's go back out of this and move into a demonstration with our dynamic DNA nucleotides. So how does the image that we were looking at before orient us to here? First, we've got our carbon at the top. This is our sugar. Here is this is an oxygen. Here's carbon number 1, which is attached to our nitrogenous base. Carbon number 2, which is attached to just a hydrogen. We've got carbon number 3, and carbon 3 is attached to that OH group. So this is the 3 end of the nucleotide. Here is carbon number 4, and then our chimney, carbon number 5, which is attached to the phosphate group. So you'll notice that
even for me to show you all of the carbons of the sugar molecule, I've got to rotate the molecule because it's not flat. It rotates in space. It's three-dimensional. So how do we go about switching this to a ribonucleotide? Well, we start here. We start with our oxygen. Here's our carbon number 1. The change is on that carbon number 2. I've got an oxygen here, so this is actually representing a hydroxyl group. If I put that hydroxyl group on the molecule here, I end up with a sugar that has 2 hydroxyl groups on it. Here's carbon number 2. I added that hydroxyl group. Carbon number 3 already had a hydroxyl group on it. And that's all it took. This is now a ribonucleotide. And let's look at some of the others. So here's guanine. I've added that second hydroxyl group on carbon number 2. It's also a ribonucleotide. Woo! We have our third nucleotide, which is cytosine, and you can see here, get it oriented, that I've added this OH group on carbon number 2 and the one on carbon number 3, so it is also a ribonucleotide. Now, let's look at the last one, and that is thymine. And thymine also, wait, did you just say that thymine doesn't exist in RNA? We'll have to address that, won't we? Maybe more than one change. All right, so let's look at thymine a little closer. Here is our oxygen. Here is our first carbon with the thymine that you just told me doesn't exist on a ribonucleotide. Here's our carbon number 2. So I'll add my OH group here. And so how do I deal with this? How do I make this into a uracil? It's a thymine. All I've got to do is pull off this methyl group. Boop. There it is. And so now we have a uracil nucleotide just by removing this methyl group and adding on this hydroxyl group. Cool.
So what's another nucleotide that we spend a lot of time talking about in biology, but focusing just on one region? That would be ATP. So let's look at how we can use this model to address cellular energetics.
And generally, we take our students' attention to the 3 phosphate groups, but I'd like for us to focus for a minute again on the sugar. And what do you notice about that sugar? Is that deoxyribose or is that ribose? You're right, that's ribose. And it's important that it's ribose. These are less stable and more reactive functional groups than the simple hydrogen that you find on this second carbon for deoxyribose. And so if we wanted to use our nucleotide for adenine and convert it into ATP, that's fairly simple to do. I'm going to come back here and show you again our nucleotide. So we have our adenine base. We have our deoxyribose sugar, which I'm going to convert to that less stable ribose by just adding the OH group right there. And now we're going to focus on adding some additional phosphate groups. All right, so I'm going to add phosphate here. Now we have 2 phosphate groups. I can spin this around a little bit. And I've got my ADP. Add 1 more phosphate group on here. Here's our last phosphate group. Now we have adenosine triphosphate. Again, not flat and moves around in space, all right, which is more accurate than our flat model. gives our students a better idea of how this works. So what do I do then? I generally have my students build these in pairs, which is helpful, but then we really need to talk about why ATP is used as the energy currency or one of the energy currencies in our cells. What happens when something is phosphorylated? So at this point, I like to do a lot of turn and talk. I would have a turn and talk activity where the partners who built these, one student would take the molecule, pop off that last phosphate group, and phosphorylate their partner. All right, so Dan's over here. I'm going to phosphorylate Dan. And what happens when Dan takes this phosphate group? Well, Dan would need to come back and tell me that phosphate group might make him a little less stable. He's feeling less stable and more reactive, or maybe it would cause a shape change that makes him more likely as a molecule to react. So that's one of the ways that I use the dynamic DNA nucleotides to demonstrate cell energetics.
Now that I'm less stable and apparently more reactive, I'd like to take a step back and go back at the central dogma. You see how there's lots of different ways that we could look at these models, but what they're really good is teaching DNA structure and function. So you don't always want to go into incredible detail. You don't always want to be able to go into all the different chemistry parts of this. Sometimes you just want your students to be able to wonder what's going on. And so Karen has, she's going to be modeling what my students would do. And I'd like to kind of take you through how I walk my students in without the terminology, without the chemistry. These would be intro bio students. We'd throw the models out and we'd say, okay, pick them up and see what there are differences. And so the students would say that, well, there's kind of this orange-red region. And then there's this kind of gray-blue region. And it seems to show up in each and every one. So we could talk about how the nucleotides have different subunits to them. Then they might start to say, well, on these blue regions, there seems to be some areas where there's, kind of circle-y type things, and I would call them rings, that we would have maybe some that have 2 circles. And then you might have in some of the class say, but wait a minute, mine has 1 circle to it. And Karen had mentioned before about the student modeling kits. We also have the foam kits, which again, we've tried to make sure that you can see there that the red one has 2 kind of circles to it. And then the yellow one has only 1. And so we could make comparisons between these things. When you're looking at the student modeling kits in our hands, you'll see that there are some arrows and there are some holes where the arrows are going to go into. And again, some students might notice that sometimes we have 3 arrows and sometimes we have 2. Of course, they are going to start putting them together. So we see that there's differences. If you take a look at the dynamic DNA, you'll notice that there's magnets on the edge. And they'll say, wait a minute, mine has 2 magnets, and somebody else might say, mine has 3. And as Karen was already showing, they're going to play and start trying to put these things together. And that's awesome. Because what you see right now is Karen put together a molecule, but it didn't seem to go together quite right. And that's fine. In fact, I'd love when the students make mistakes, because then we could start talking about, well, do we have to fill up all the magnets? What do you think is going on with this? Sometimes they'll put the phosphates in with the areas where the hydrogen bonds will be. There'll be all sorts of mistakes. And we could start discussing what kind of things would be built. So at this point, the students would be putting together the 3 magnets with the 3 magnets, the 2 magnets with the 2 magnets, or the 3 arrows with the same within the foam. And I would ask again, what do you notice? And when you put all these together and you lay them down, they all wind up being the same size. And the students would be, it's always a pair of 2 with a single. So it's a pair of 2 rings with a single ring, and it always winds up being the same size. If you hold up the dynamic DNA, you would notice that there's kind of a flat region along the middle. And then you would have these areas on the side where they would be sticking out. Now, the other thing that you would start noticing is students are going to try to put these base pairs together. And Karen jumped ahead with the whole helix, but the students would put them together one at a time. And they'd say, wait a minute, we can start getting this kind of ladder-like structure that's out there. And then from that ladder-like structure, you know that somebody's going to start twisting it. And as they start twisting it and turning it, they realize that it's turning into a helix. And that's when the students get really excited in the classroom. And I have the, no, quiet down. Let somebody else discover it on their own. And of course, they start putting it together. Karen had a piece before that I think was 6 pairs or 8 pairs. If you hold it up, you can start really seeing the double helix structure that we would have in the classroom. And they'll notice that all those red and yellow things are on the outside, and all the blue and white things are in the middle. And they'll notice other things about the structure. Notice I'm not using any real language here. What do you notice? What do you wonder? What questions do you have? What are you curious about? And they can play. We can do the same thing with the foam models, where we can put those into the gray area. Karen showed in her video before that it has the little arrows of where everything goes in. And again, the students will instantly see double helix structure. Aha, they know what that is. When the students discover the structure on their own, this is where the power of this model comes from. It's not that I'm teaching vocabulary. And I can always double back and say, remember the red and yellow parts. That's the phosphate groups. Remember this, that's that. But they will always have the play in the back of their minds. The other thing that makes a really, really good model is not that the students can just play with it, but as they're playing, they start to generate questions. And the questions that you're going to see here are not questions that I came up with. In fact, I say this in the video. These are my students' questions from just playing with the model and seeing what's out there.
Karen and I said it right at the beginning that what makes dynamic DNA such a great model is some of the questions that it promotes. And so looking up on the screen right now, you see how the dynamic DNA is twisting and turning, and having the students play with this brings up questions. Can it compress around the nucleosome? How does it compress around the nucleosome? Do you have some nucleotides are getting closer, some are going to be more exposed? Is the sense strands going to be something that's more on the outside of the nucleosome or the inside of the nucleosome? Is there going to be any steric hindrance that's going on within there? Can we elongate the molecule? Is that how expression occurs in some cases, where an enzyme will come in and pull it apart or maybe compress it down? How do these forces work within the actual DNA molecule itself? Well, we can model it and we can see how these things are going. The second thing that we would look at and see is RNA. We can build the RNA, and Karen showed us how we can put all these things together. Well, I made a little bit of a chain of RNA here. And in this chain of RNA, take a look in this spot here. You can see all these oxygens in a row. That's going to make it really unstable. That's going to be a problem. It's one of the reasons why RNA is not around for as much. And so then it brings up the question, well, then how do we come up with all these major structures that are made out of RNA and the three-dimensional things that are going on within there? So this is chemistry questions that can come up. And then we get to the really interesting stuff. Now, what I'm about to show you is stuff that I was modeling out for my students, and then my students started asking these questions. So these aren't my questions, these are my students' questions. So we see that we have a replication fork, and I put an RNA primer on there because we're always talking about an RNA primer, and I could model it with dynamic DNA. And we have the free 3' hydroxyl. And you can see the cartoon image of the free 3' hydroxyl up that we show in so many of our models. Well, why is it important? What's going on with that? Because that's where the new nucleotide can attach. So the replication fork pulls apart, and we could see DNA polymerase coming in. Now, this is a little bit corny in the way that I have it modeled, is the DNA polymerase would be clamped on and would be working with this, not something that's marching in from the left, but it helps the students to understand. And so we see the DNA polymerase marching in, and you can see all the phosphates now are getting really close to that free 3' hydroxyl. So that hydrogen on the free 3' hydroxyl is going to be important, and all those oxygens in the phosphates are going to be important. And we can see exactly what's happening at the active site right now of DNA polymerase. And as the active site pulls it in closely, you're starting to have the adenine matching up with the thymine, and those The phosphates are getting really close to the free 3' hydroxyl. We could see it here as they're kind of overlapping with each other. So what's happening? Well, you could also see that in the way my model is it's causing the nucleotides to kind of bend and move a little bit. Was that real? What's happening? What's the chemistry that's coming on? These are the questions that my students were asking. And so it brings out a little bit of wonder. It's okay if we don't have all the answers on this. We want our students to say, hey, whoa, wait a minute. Is that real? Is that what we're looking at here? And so I pulled it apart a little bit so you can see the interaction. And of course, at this point, we pull off, and then my students were like, well, what do you pull off? Do you pull off a monophosphate? Do you pull off a diphosphate? And I was like, oh, I don't know. In my whole teaching career, I had always said, well, it's a dinucleotide triphosphate. It comes in, it creates the covalent bond in the backbone, which we can see this covalent bond forming here. And we can see that I pulled off a hydrogen and I've got a diphosphate. I had no idea that that actually happened. My students were asking me that question, saying, well, what does happen in that case? And I was searching for answers. So I had to go back and do a lot of literature reading. And that's okay that I didn't know that answer, that my students came up with that question. My students were asking the question because we had done ATP and we had done glycolysis, and everything was about, well, if I phosphorylate this, where's the hydrogen going to go? Where's an oxygen going to go? Where are all these things going to go? And my students were looking at the same way, cross-cutting concepts. This isn't just DNA, this is biology, and everything works together. And then, of course, it turns out it is a diphosphate that pulls off. Whoa, who knew? I sure as heck didn't. And apparently the hydrogen, it becomes an acidic situation. And so the hydrogen is free within the solution. So where does the phosphates go? What happens with this? And you'll see here that I brought in a new enzyme that I learned about called pyrophosphatase. And pyrophosphatase is going to come and break apart those two phosphates. And this is a highly negative Gibbs free energy, which is going to help drive the reaction. So you don't have to go into all this. I would never do this with my intro bio students. I would want to see my AP Bio students start questioning about this. And now that I could bring up the huge negative Gibbs free energy change, we can start talking about this in the same language that we talked about within cell respiration. Cross-cutting concepts. How is this all working together? What are we working with? This is what makes this such a great model, because it leads to more questions and more questions and more questions, and that's what we want to see.
So very interestingly, those questions were coming out from, in a sense, 2 years ago, because I worked with my students, we played with the models, and then we put this presentation together. And then since then, COVID happened. And at this point, you might be watching this and saying, oh, I can model something like this up on a screen, or we could show a video or whatever. The questions were not generated when they didn't have the models in their hands to play with. The questions were not the same this year when I was showing things virtually up on a screen, or I was just demoing something. The questions came from their holding on, touching, twisting, turning, playing with everything. It was much better to have the models in their hands. The second thing I just want to point out before Karen hops back in is that we can actually show all of replication, transcription, translation using different kits that 3DMD has. So you can show the entire flow of genetic information. We're not doing that here today for time constraints.
So one of the cool things that they've done at 3DMD to support their student modeling packs and also their full-scale kits is to make companion activities that are digital. And these are available, I think Rachel just put a link in the chat for that. I want to give you two ways to use these. First is to help scaffold a transition between models. I think a lot of times, at least I've caught myself assuming students understand that the ribbon model of DNA, what that means when we transition to the actual molecular model, but they need that scaffolding. And then Dan's going to talk to you about how he uses this for assessment.
One of the nice things that is available on the 3D Molecular Design website, where you find these kits, is a set of digital activities that go with each of the student modeling packs. This one slide has been taken out of a collection of slides that come with the student double helix modeling pack. And I'm going to use it to talk about moving between models. It's really important that we don't assume just do this on the fly. We need to either allow them a time to puzzle it out or deliberately, depending on your situation, make these connections between the different models. So I'm going to do so right here. I am going to take a look at the textbook model, the one that shows us where almost all the atoms are in relation to each other in two-dimensional space. And we've got our dynamic DNA nucleotide here. We have a nucleotide from the student modeling pack that you've received. And this nucleotide isn't a big part of our presentation, but you'll find this in our flow of genetic information kit. One thing you can do is actually click on each of these labels here, and we can move them to where we want to on the slide, and they're layered. So I'm going to say, okay, so this is our nitrogenous base. We have a purine here. And if we were looking at the same area on the dynamic DNA model, it would be here. This is a guanine nucleotide. And so we again can see our double ring structure here. When we move down to the double helix student modeling pack nucleotide, the nitrogenous base does not show every single atom, but it does show the double ringed nature of this purine. And in addition to that, you can see that this guanine nucleotide is going to form 3 hydrogen bonds. You can see that on the dynamic DNA model, but you can't see it as specifically here unless you really know your biochemistry. And then lastly, you can show the nitrogenous base here. The structure of guanine is further de-emphasized, and the focus is on the bonding in this model. So next, we would look at the backbone. And here, again, you can see our phosphate sugar backbone in our textbook model or our flat diagram. Here, again, is our phosphate and sugar of the bone. When we go down to our student modeling pack, the sugar and phosphate backbone actual structure is de-emphasized because we want to focus on the nitrogenous base. And yet we still have a couple of cues here, especially at the end of the ribbon model that this represents. It's got a little point on there. So that's going to be suggestive. We'll address that in a minute. And our sugar phosphate backbone here on our flow of genetic information nucleotide. And finally, remember that we talked about how DNA polymerase and RNA polymerase add new nucleotides during elongation to the 3' end. And so we talked a little bit about that. So let's identify our 5' end of this model and our 3' end here of our flat textbook model. And as picky as you want here. Or not. Too picky. Let's just do this. And we've got our 5' end of this nucleotide here and our 3' end of the nucleotide. And so we're going to be adding new nucleotides to this portion of the dynamic DNA model. Here we have our 5' end of our student modeling pack and our 3' end. And finally, the 5' end of the flow of genetic information kit nucleotide here, our 3' end here. And of course, you want to have further discussion on those things with your students. Another nice thing about these slides is that we have editable text boxes here. So you can have your students type in their answers here. and then save the slide and share that slide with you for assessment purposes, formative or summative, your call. But in addition to this, as an instructor, you could put your own question in here. Maybe you don't want that. Maybe you want some different type of information out of them. So you can resize this box. You can retype a different question in here. It's up to you how you use this, but it can be very helpful either in a face-to-face situation or in a situation where you are distance learning.
So as Karen mentions before, that I use these as an assessment tool because I have found that the students will get real excited about the models and they're having so much fun playing. And I'll get kind of an impression of the students who are very vocal, that they're getting it, that they understand. But some of the students who are a little bit more quiet, I don't know what's going on. Or maybe even the vocal ones are vocal, but they're still not getting it. With something like this, these are all drag and drop. I can pair the adenine with the thymine, both in the molecular structure and within the foam structures. I used to have students take pictures of the models and try to label different things, but now I could actually have them just drag and drop these and use some of these manipulatives to figure out, are they getting it? Where are some of the misconceptions? Some of the base pairs came out really weird when I had the students do this after they played with the real thing. Now, finally, I teased a little bit in our introduction that one of the things that makes us such a great model is not only can it inspire questions, but you can see so many things with it and teach structures in ways that you hadn't thought possible before with all the twisting and such. But it's also a scale model. And as a scale model, I've been taking photo 51 from Dr. Rosalind Franklin. And if you read her documents, if you read her data, you could actually go against the claim of Watson and Crick's model. So I have over here the dynamic DNA and a metric measuring device, a ruler. And I've had the students where they would be able to go through and look at the rise between the different base pairs and then compare it to the rise of the how many angstroms you would have on Dr. Franklin's image. And then you could talk about the symmetries that are going on here and all the different things that her work puts out. So instead of just having, oh, Dr. Franklin, had this wonderful picture and it was just passed over and Watson and Crick made this model, we could actually look at Dr. Franklin's data versus the claim of the model. And now we're giving her the due that she is deserved because she was an amazing scientist who had amazing data as opposed to just interpreting that data and seeing where the model went. And I've been playing around with it. It worked pretty well. And it was a great way for students to get the story of her amazing work, also the structure all at the same time without talking about a photo that didn't mean much to the students before. And do we have any questions?
Well, thank you, Dan and Karen, for a wonderful presentation. And if you guys have any questions, please post them in chat. This is also the time when you could unmute and just ask your questions directly to Dan and Karen. So while we're giving you a second to compose your thoughts, I will just want to remind you about some of the things that happened in our session today. So Dan and Karen were talking about our dynamic DNA kit and the double helix student modeling pack. And so we have those two materials available. Rachel posted the link to be able to get your free double helix student modeling pack. And I hope that you have had a great conference and that you will be able to catch all those other sessions on recordings or whatever is in your future to wrap up NSTA Engage. So